Okay, what we're going to do in this lab is we're going to use dialysis tubing to simulate a cell that has various solutions in it, and we're going to put that in water, and we're going to see if we can determine the molarity of the different solutions from the change in mass. So we've got a bunch of colored sucrose solutions. We don't know which concentration is which, so we're going to put them all in dialysis bags, put them in water, and see after about 30 minutes if there's a change in mass or not. So to start, we're using dialysis tubing, and dialysis tubing looks like a piece of tape with no sticky stuff on it, but it is in fact a tube. So what we have to do is we have to soak the tubes in water. I actually have 6 tubes here. I'm sure I've got 6. We're going to soak them in water, and that allows it to soften up so we can actually open it up and form a tube. So what we have to do first is we need to tie a knot in one end of the tube, and then we can rub the other tube between our fingers and it will open up the tube. And then what we want to do is we want to use a funnel, and we are then going to fill the tube with approximately 15 milliliters of solution. And we're going to use each of the different colored solutions. Okay, so I'm going to start with the red color, and I'm going to just measure out 15 milliliters. Tube. And then we want to leave some room for water to enter into our tube. I'm going to just place that down there, and then I'm going to fill the next tube. Rinse that out. And then I'm going to dry them and weigh them. So it's going to take a minute to do that. So again, I'm tying a knot on one end. Okay, so now I have filled all the bags. So I'm going to dry them and weigh them and record the mass on my worksheet here. I'm going to start with the red. So that first one, so that's red, initial mass 18.51 grams. And we're going to put that into a cup. Take orange one and mass that. So orange. And that's 19.86 grams. Into a cup. Got the yellow one, which is zeroed. And then yellow, 17.63 grams. Put that into our next cup. 18.94 grams into our next cup. And then we've got our blue. 18.44 grams. That into a cup. And then our last one. And then purple. 19.10 grams. All of our cups, all of our different colors. I'm just going to fill them with water. And what we're going to do is we are going to just let them sit for about half an hour. And then in half an hour, we will take them out and we will mask them again. So just letting them sit for half an hour. Okay, our samples have been sitting in the water for 30 minutes. So what we're going to do is we're going to take them out, dry them, and then weigh them again for our final mass, and we're going to see if we can figure out from the initial mass to the final mass if they have increased or decreased in mass, and then what the molarities of the different colored solutions are. Okay, so let's start with the red. Out. Dry that a little bit. And record that mass. So final mass 20.94, and so we can put that one out. Just gonna dump it out so it's easier. Orange 23.50, that one. Got yellow 17.75. Green 24.32. Blue 23.61. Purple 20.52. Next, I want to just quick point out this is gained significantly because it's very hard and rigid, which it wasn't before. So that should tell you something about the blue one. So we can kind of take a visual check compared to what we started with and see just by feel if any of them seem like they have gained mass. So that one definitely does. So that should be a clue to what its molar concentration is. So what we want to do now is we want to use that data, and we want to try and figure out if we can use that data to determine the molarities. We probably should calculate the percent change in mass so that we can compare better our sets of data. So we need to do a little bit of math and a little bit of calculations using our data to see if we have a change in mass and if we can calculate our molarities. So we will do a little bit of math to determine that.